Welcome to our training on the primary characteristics of sterile dosage forms. This training will provide you with a comprehensive understanding of the essential characteristics that must be met in order to produce safe, effective, and sterile dosage forms. We will also cover the concept of osmotic pressure and tonicity, looking at how they play a role in maintaining the integrity of cells and the proper functioning of certain organs in the body. The importance of isotonicity in sterile dosage forms will also be discussed, as well as the different types of solutions with different osmotic pressures. And we'll end the session with a general overview of the quality tests applicable to parenteral drug products. Okay, let's get started. There are 7 primary characteristics of sterile dosage forms: safety, sterility, freedom from pyrogenic contamination, freedom from visible particulate matter, stability, compatibility, and isotonicity. Let's talk about each characteristic one by one. Sterile dosage forms are injected directly into the body. This means that they bypass the body's natural defenses against harmful entities, making safety a crucial aspect of their formulation. During product development, any component of an injectable product must be thoroughly tested and proven safe at the quantitative level at which it would be used. Sterile dosage forms must be free of viable microorganisms, including bacteria, fungi, and viruses. Sterile dosage forms must be free of pyrogens, which are substances that can cause fever or other systemic reactions. Sterile dosage forms must also be free of visible particulate matter, such as bits of debris or fibers. This particulate matter can cause irritation or blockage of blood vessels or other body parts. For example, ready-to-use solutions and solutions that are used for reconstitution must be free from any visible particulate matter and must meet established standards for the number of subvisible particles, which should not be larger than a certain size. The standards for particle size vary across different compendia, but in most cases the maximum size is 10 microns and 25 microns. Sterile dosage forms must be free of microorganisms. They have to maintain their physical and chemical characteristics over time. Any degradation or contamination of the dosage form could compromise its effectiveness and potentially pose a risk to patients. Many sterile dosage forms must be manipulated before injection. For example, some need to be reconstituted by sterile dilution or combined with another solution for administration to the patient. Therefore, the sterile dosage form must be shown to be compatible with diluents for reconstitution or diluents for infusion. The same is applicable when 2 or more drug substances are combined in the same dosage form. Isotonicity refers to the similarity in osmotic pressure between the solution and the body's fluids to prevent damage to cells and tissues. Isotonicity comes from the term tonicity. Tonicity is a medical term that relates to the osmotic pressure difference between the internal and external sides of a cell membrane. Osmotic pressure plays a critical role in maintaining the integrity of cells and the proper functioning of certain organs, and it is important to consider it when formulating sterile dosage forms.